DNA replication is a complex process that includes various steps. Before we can learn about this process, there are some key facts you must understand first. DNA consists of a double helix that contains genetic material. It is made up of a nitrogenous base, sugar called deoxyribose, and a phosphate group. Within the base, one can find adenine, thymine, guanine, and cytosine. Chargaff's rule states that the value of adenine in a strand of DNA is equal to thymine. Guanine also equals the amount of cytosine. DNA polymerases are responsible for elongating DNA strands at a replication fork. When nucleotides appear at the template strand, DNA polymerases add them to the growing strand, also known as 3'. Nucleotides are always added to the 3' end, since the overall direction of replication is from 5' to 3'. The copying of DNA occurs in a process known as DNA replication. Let's take a closer look, starting at the origins of replication.
To begin DNA replication, two double helix strands are unwound by the helicase enzymes. The point at which a strand is separated, as well as where new DNA is synthesized, can be called the replication fork. Single-strand binding proteins, or more commonly called SSBs, maintain the separation of the DNA strands throughout replication by quickly covering the new strands that were unwound by the helicase enzymes. The SSBs bind very loosely to the DNA, and the polymerase enzymes begin to create the new DNA strands. However, without the SSBs, the DNA strands could easily bind back together after being separated. The double helix structure of DNA consists of two anti-parallel strands, which means the complementary 5' to 3' strands go in opposite directions. Since the chain synthesizes in the 5' to 3' direction, polymerase 3 then attaches to the nucleotide at the 3' end and hooks the 5' phosphate group of the incoming nucleotide onto the 3' hydroxyl group at the last part of the growing nucleic acid chain. Even after the DNA strands are separated from one another, the DNA polymerase cannot begin doubling the DNA immediately. The DNA polymerase can only extend a nucleic acid chain. It cannot start from scratch. Therefore, to give the DNA polymerase a beginning point, an RNA polymerase called primase synthesizes the RNA primers, which are complementary strands to the DNA. Now, DNA polymerase is able to duplicate the DNA strand. Next, the DNA polymerase synthesizes the complementary strands, so there is one for each of the two parental strands. The polymerase on the leading strand is able to stay on its DNA template and duplicate the DNA into one continuous strand. However, the other polymerase on the lagging strand is only able to copy a short part of the strand before it collides with the primer from the previous sequence portion of the DNA strand. That polymerase then has to release the DNA strand multiple times and go further up the DNA strand to release the RNA primer.
The continuously synthesized strand is called the leading strand, and the strand that is synthesized in short pieces is called the lagging strand. The DNA pieces separated by RNA primers in the lagging strand are called Okazaki fragments. Before the lagging strand DNA can leave the replication process, its RNA primers must be removed, and the Okazaki fragments must also be joined together in order to create a continuous DNA strand. First, the RNA primer must be removed from the lagging strand. The RNase H then uses RNA and DNA helicases to degrade DNA primers. Then, the gap that was created by the RNase H is filled in by the DNA polymerase. The DNA polymerase extends the Okazaki fragments. Finally, the Okazaki fragments are joined by DNA ligase, and the fragments are ligated. The 3' end then connects to the 5' end of the lagging strand, and there are two new DNA strands created. And finally, the process of DNA replication is complete.